Hey, all, there's still lots of seats, so if you're standing in the back, don't be shy. Come and sit down and join us.
Okay, thank you. And there are indeed seats up here. So I'm Louis. Oh, I think this works better. I'm Louis, and I'm presenting work that I did mostly when I was at the MBL with Per Borg's group, although I've now moved to China to start my own group. So, and I already saw some people taking pictures of slides, so, why don't you just take one picture of this one and you get all the slides online? And that's, because they're all, actually I'm even presenting from this public URL. I'm even going to refresh so I make sure I have the latest version. Okay. Yeah, okay. All right, so, oh, sorry, okay. I don't really have an introductions because I figured that you can just pretend that I said exactly the same things that Devin and Alex said at the beginning of their talks. And what we're trying to do is get an eierlegenden Vollmilchsau. Okay? And here our research question was, okay, if we're willing to sacrifice generality and focus just on bioinformatics and focus just on the kind of problems that we were interested in, can we get a better tool? Can we and by using a domain-specific language? So in the previous talk, we already saw this mention of the Nextflow domain-specific language, which is very powerful, can do a lot of things. So here the trade-off is, can we be less powerful but do those few things better? And what does it mean to be better? So the number one thing, we want to have correct science. We want usability, reproducibility. We want things to be understandable. We want to support best practices because, and again, going back to the previous talks, there's a lot of documents out there saying this is what you should do. And nobody will actually disagree with you that that's the best practice. But that doesn't mean that people actually do it so we want to lower the activation energy to do that. And finally, we also want the tool to be fast. Okay, so this is sort of the eierlegende Vollmilchsau slide. So, on the one hand, usability, even usability we want things that work out of the box. Bioconda install, run pipeline with single command or a single click, and that even people who are experts in their biological problem, they should be able to use it. We also want things to be really flexible and customizable and be able to tweak it to our own very specific problem, which nobody else in the world has, because my problem is special. And people who are not afraid of the Unix command line, they also want to be able to tweak. And so even the word usability, you're already pulling stuff apart. And in science, we also, projects can last a long time. We want things to be reproducible. We want to now be able to reproduce a project that was published a couple of years ago that maybe had data from a while back. And at the same time, things move. What we were doing, maybe there's new methods out. Stuff moves fast. So our solution, this is what I'm proposing, is this domain-specific programming language called NGLess. And so here, actually, this is a complete script. So we don't need to go through the whole thing. I'll just point out a couple of things. So here at the top, so you declare everything's version. So you declare version. So we just released version 1.0. So we actually had a talk yesterday encouraged us to call things 1.0. And we synchronized it. So the publication came out last month. And as we knew the paper was going to come out, we said, OK, this is version 1.0. But then everything else, and there's a couple of modules that you can introduce that these add things like novel functionality, novel data sets that are integrated, and you also version that. And then everything, it kind of looks very Python-esque. We also stole some ideas from Ruby. And here basically you're loading some samples, some set of FASTQ files, doing some pre-processing, then mapping, and then doing some summarization analysis. The most frequently asked question is, why are we doing this? If we wanted to have something that looks like Python-esque, we could have just used Python and developed a library in Python that that basically did this, or there's other projects for workflows already out there. And so I'm going to try to spend the next 12 minutes trying to show you the advantages of this approach. So so this was maybe twice as much effort as that would have been, but I'm going to show that, okay, at least this is what we get out of it. So just as a programming language, so this is a very standard imperative style programming language. You read it from top to bottom. Everything is statically typed, so the types can all be inferred. And as I said, it has very limited functionality. You know, if you want to get technical, it's not Turing complete. You cannot define your own. You're limited in what you can do. You can only really do the stuff. You can only really do stuff related to bioinformatics. But it is adapted to bioinformatics, so things like short reads, etc., SAM files, they're all concepts that the tool itself knows about. And so what are the big advantages of this? So one of the things, and we actually spend a lot of time on this, is that there's a lot of tools that work really well but fail really badly. That means that if you give the tool everything it sort of needs and wants, then you get your results very fast, good results. But if something's wrong, you know, then you get some sort of error happened, you know, core dump. And so actually I don't have I've tried Google Scholar to find one to get a proper citation for this claim that most time that you spend on software development is debugging. But I think if you've written more than one single line of bash, you know, this is like getting a citation for water is wet. I mean, yes, we spend most of our time trying to figure out why stuff is going wrong. And so, you know, it's like when you're optimizing software. You want to optimize where the software spend the most time, cut that down. Where do people spend the most time? Let's try to cut that down. So, you know, bad error messages, we treat them as bugs. That's actually, and actually, please report them. I think there's sometimes a bit of a feeling of, it's like, as a user, if you get core dump, that's like a bug in the software. You know, you should report that. If there's an issue, the software should tell you what the issue is. If it's run out of memory, it should say, I've run out of memory. If your file is malformatted, it should say line 317, that's an error. So please report bad error as bugs. You know, we try to fail clearly. So this is actually a, if you don't know it, this is a parody account. But obviously the joke is that this happens way too often. And actually one of the things that often happens in many tools is so the tool starts writing its output, and then, you know, somehow something goes wrong, and then you have a half-finished file. So that's the type of thing that we don't do. So we try to, and this is really enabled by the fact that the language is very limited. So a lot of things can be done to infer what the user really wanted to do, as opposed to, you know, if you're trying to do this where you're dealing with, say, a bash script, it's impossible to control all of the possible things that can go wrong. And for example, one thing that we can check is, you know, do the input files exist? And, you know, do the output directories exist? Do you have permissions to write for them, et cetera, even before, you know, and we can do this as soon as possible. In some cases, that could be really trivial. So here's a very trivial example. So again, just to give you a feeling for the language. So, you know, declare all versions that you want. And then, you know, you're going to read from this FASTQ file, just a single file, and then you map it. And so here, if you don't specify anything, it's going to use BWA, although you can, if you just add comma mapper equals minimap2, it would use minimap2. Right? But here you're mapping to a known reference that comes from this module, and then you're writing to this file. And here the file's right there. So it's actually quite easy. It will be quite easy to replicate this in other settings where you can just check whether all of these paths are fine, and then, you know, before you even do the mapping. We actually can do a little bit better because we can, you know, so in this case, also trying to, so once you have many samples, one way that we've used to organize this is you sort of do an indirect where you keep a list, you keep a file with the list of your samples, you're called samples.txt. And then you index it by a job index, and And you run this multiple times and doing it even in parallel. And then you preprocess each one individually. And now here, the problem is you need to get halfway through the script to know where the latter part of the script is going to use. But here we can sort of analyze the abstract syntax tree and move those checks and try and still do those checks as early as possible. So as early as the language knows, oh, in 5 steps I'm going to need to use this directory, can already go and check, oh, does this directory really exist? Does the directory have the right permissions, etc. So a lot of this is this type of really small things, but in the end, this is about providing a better user experience. So as we saw, I think in the previous talk, we can annotate the outputs. We can annotate the outputs so that here we print as comments in the output, or this depends a little bit on exactly what the output format is. But so here, if you just have a text file, then it outputs as a text file, both in a way that's nice for humans to read, and also in a way that's nice for machines to read. Here there's a couple of other tools that then can use this to check, okay, were these things computed in an equivalent manner, yes or no? Okay, so as I said, everything is versioned. So we can use this to evolve the language and the APIs while keeping backwards compatibility. So already we have some things that work differently in version 1 versus the previous versions. And again, as in the previous talk, one thing we always keep in mind is, so this script should completely define the computation. So it's from the computer science point of view, the output is a deterministic function of what the inputs were plus the script. So and there's a lot of incidental information, and we've all gotten these scripts from someone else that refer to paths in their home directory. We've all gotten scripts that refer to something that exists in their cluster environment. And so here, all of those things are really important, but here we've separated them so that so those things are, you can specify those in a configuration file where and you can tweak a lot of these things, but they're not part of the actual script because this way you can share the script. I mean, we've started doing it. We have now a couple of papers where we've done this, where we've published alongside the paper saying this is what you used. And in principle, someone takes that, they run it with their own system, but as long as they use the same data and the same script, they should get the exact same tables as we had. Okay, so as I said, we've recently gotten to version 1.0. We have a set of built-in functionality, mostly handling FASTQ files, processing them in basic ways, so we always do QC. You don't specify that you want to do QC. You don't get a choice. Any operation that modifies a file will get QC at the top and QC afterwards. We also benefit from, this is sort of done in a streaming manner, so that as stuff is being read from disk, it gets QC'd in, and as it's being written back to disk, it gets QC'd out. You can also extend it. You can always extend it just by writing code and contributing code. That's one way. Another way is, so if you're just adding basically a function that takes some inputs and produces some outputs, you can do that with a YAML file. So when we have a couple of those already. So just to give you an example of the type. So this is an actual module that we have. It's called Motus from Alessio, who's sitting over there. He's the first author there on this tool. And so here in this YAML file, you specify what that it's going to take a read set. So that means it's going to take a set of FASTQ files. And here it says that their tool can handle if the files are gzipped. And then NGLess can then handle all of the conversions. So if you give it a file it's in bzip2, then it will get auto-converted. So all of those things are the type of things that we think the users shouldn't care about. They should think that conceptually, I have a set of reads. This tool takes a set of reads. I should be able to plug this together. And these things, and all of the things, and so for example, one thing that this tool cannot do is cannot stream the reads. So that means that sometimes we might need to do a do a temporary file. But the users really shouldn't care about these things. And then one thing also that's important is, so here at the bottom we have the citation, so that whenever you use this, we print on the command line that please we should cite Alessio's paper. Because that's actually a problem, that sometimes things get integrated into bigger tools, and sort of the identity of the little parts gets lost. So at least we try to support the best practice of citing things by printing it out for, and we hope that the user can at least copy paste that into their paper. So this is what I was saying. So best practice should version all the things. Rarely really follow through. It's very hard to version, because in principle you'd have to version all your stack. So here at least we're trying to just version a small number of things, make it easy to cite. Quality control, we always do that. And as I said, we isolate the system-specific information. So we've done this for metagenomics. So we've developed a couple of pipelines for different environments. So this was not a novel approach. The approaches had been done before. We actually had an earlier tool called MoCat that did a lot of this. And we just re-implemented it with NGLess. So as I said, we never want to say we should use it because it's fast. That's not the most important thing, but we're still really fast. And in particular, we're really fast because, so a lot of the other tools, so we tried htsec count as a comparison. I actually tried to use feature counts. It wouldn't run even with 1 terabyte of memory. And that's just because these tools were done for quote-unquote small things like the human genome, whilst in metagenomics, we really have much larger data sets. So we actually scale much better to these larger things. So I'm coming to the end. So the slides are open, are public. So you can install this with Conda. It's a single command. So 11,000 people have done it. So they can't be all wrong. We have a paper that really came out last month. We have what I think is very complete documentation. All the code is on GitHub, and we have a mailing list. So and so, to summarize, we have a domain-specific language. So here, as I said, there's this trade-off where we've sacrificed a lot of generality compared to other solutions, but we're trying to give the users a better experience. We've gotten to version 1.0 just a few months ago, and we're focused on metagenomics. I actually know of one person in the world that has been trying to use this for RNA-seq. I think it would work. It's just a bit outside of my own and our own personal expertise. So with that, I want to thank the other collaborators and also all of the users who reported bug reports. That's actually really nice. It's really helpful that you guys help with that. Actually, some people in the room have reported bug reports. Thank you. Praise is also really good. And also thank you for everyone.
So I don't see any questions online. If anybody in the room wants to ask a question, the microphone's on this side. All right, seeing no questions,
Okay.
Thank you.
Thank you.
All right, coming up next, we have 3 short talks, so we will be saving